This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 128, recorded April 8th, 2011. Hi everybody, I'm Vincent Racaniello, and this is TWiV, the show where we talk about all the latest news in the wonderful field of virology. And joining me today from Western Massachusetts is Alan Dove.
Pleasure to be here.
How you doing, Alan?
Doing okay. How about you?
I'm all right. We have a lot of interesting virus news this week.
Yes, we do.
Looking forward to that. Also joining me across the desk here in the TWiV studio is Dickson Despommier.
Hey, Vince.
How are you, Dickson?
I'm doing well, Vince.
I haven't seen you in 5 minutes.
That's right.
Now that you are outside, I see you all the time.
Exactly.
So, are you still my friend since you see me all the time?
Yeah, still hanging in there.
Yeah. All right. Dickson's having lots of vertical farm meetings.
Indeed.
He's going vertical. Also joining us today from elsewhere in Massachusetts. It's not... Now, Alan is in... what do I call it? Western Massachusetts. So I guess this would be Eastern Massachusetts.
Oh, yeah.
Welkin Johnson.
Hello. Where am I?
Where are you?
Who are you?
Who are you?
Or as people in Eastern Massachusetts refer to it, Massachusetts.
Right.
Welkin is in the New England Primate Research Center. Is that the right name?
That's right. Yep.
Which is associated and affiliated with Harvard Medical School, and that affiliation is the Department of Microbiology and Molecular Genetics, right?
That's correct. We call ourselves the Southborough campus.
We had a colleague of yours here this week, Roberto Coulter.
Oh, Roberto.
Yeah.
Did you tape with him?
No, he gave a seminar.
Oh.
But
I love hearing Roberto talk.
Yeah,
he always works food and things into his talks.
Yeah, it was a really nice talk.
Sure.
We're going to have him on TWiM one of these days. Okay, Rich Condit is actually on the road, and he will hear this when he listens to us next week, maybe. So, hey, Rich, hope you're having fun. He's on vacation. But he'll be joining us next week. He's actually going to be in studio.
Oh, come on. The guy lives in Florida. Where's he going to go on vacation?
I think that's good. He told me his wife was complaining that it's 80 degrees and sunny, and they're driving north where it's cloudy the entire way.
Right.
And right now it is cloudy.
It is.
And it's probably in the 50s, right, Dickson?
Probably in the 50s. It's not too bad outside, though. It's not too bad at all.
No, we're not complaining.
Nope.
All right, today we have 3 stories to do.
Oh, can I just apologize to your listeners really quick?
Of course.
I've been partying hard all week so I could try to develop a Rich Condit, scratchy baritone voice. And I haven't got there yet.
Yeah, you have to do something else. I'm not sure what Rich does, but he hasn't given away his secret. It's no problem.
Hard living.
So that means you're not going to say anything?
Yeah.
Okay. So you're too tired. All right. Now, the first story we've had for a few weeks, and I was debating whether we should do it on TWiV or TWIP, because it could be on either show.
Exactly.
We're going to do it on TWiV, because someone suggested the title, TWiV Infects TWIP. We may not use it, but I like the idea. And this one is a science paper entitled Leishmania RNA Virus Controls the Severity of Mucocutaneous Leishmaniasis.
Indeed.
Dickson, I got the accent right on leishmaniasis,
You did.
right?
You did.
That's because we did a TWIP on it,
This is exactly right.
and that's TWIP number 14,
Yep.
which you should check out at microbeworld.org/TWIP.
Yeah, this is not a nasty leishmaniasis.
It's not?
No. This is one of the more severe ones that really disfigures people.
This is mucocutaneous,
Yeah.
as opposed to just cutaneous.
Yeah, that's right.
So which
Or
is
visceral. Or a visceral.
So
There's
visceral is the worst, right?
Well, you can die from visceral leishmaniasis, but you can also die from mucocutaneous leishmaniasis.
So tell us.
This is a nasty one.
Yeah, but this one disfigures. I mean, the other visceral leishmaniasis extends your belly because it swells up your spleen and your liver. But to be honest with you, I would much rather have that one because they're all cured the same way anyway than I would have this one, because this one erodes away the soft palate and it attacks the mucocutaneous junctions of the urogenital tract as well. And it's an awful, awful, intractably ugly disease to catch.
And it's endemic in many parts of Latin America, right?
That's right. It's not found anywhere else except in the New World. That's right. And it's transmitted by sandflies of the genus Leishmaniasis.
These are protozoans.
They're protozoans, they're eukaryotes.
And
but inside of each one of these little devils sits a double-stranded RNA virus.
not every one of them.
No. But these that cause the severest
Right.
forms of mucocutaneous.
Oh, that's it. Story's over. Move on to the next.
No. We don't know why, though, do we?
Dickson, let me ask you.
Please.
This is an intracellular parasite.
It is.
So it's delivered by a sandfly.
That's right.
And now, if I remember, they bite you.
Yes.
And they don't inject it, do they?
No, this one is injected.
This is injected?
Yeah, this is injected.
All right.
The promastigote form of this infection lives in the invertebrate intermediate host, which is also the vector. And it undergoes stages of development inside this vector. So it's a necessary stage of the life cycle. And it's flagellated, okay? So this is a flagellated protozoan as an infection inside the Leishmania intermediate host, which is also the vector. But once it injects this into you, the mammalian, warm-blooded eukaryote host, the final host for this infection, it drops its flagellum, is picked up by macrophages, and the next thing you know, it's intracellular.
So it's carried around by the max. And where does it end up? Everywhere?
Well, it starts out as a lesion, and it looks just like the cutaneous forms of leishmaniasis.
So at the site of bite, right?
At the site of the bite, and you get this craterform ulcer, and it develops, and then it disappears, and everybody says, great, my immunity kicked in and it's gone, but it's not really gone. Next thing it could appear at the junction of the upper lip and the lower lip as another lesion. And at that point, everybody in the neighborhood would know that you've developed the mucocutaneous form instead of the cutaneous form.
We have a photo on our TWiV episode of a young lady with a lesion on her, her chin, right?
Now that's what the original lesion would look like.
She was bitten there.
She was.
I see. And then it goes elsewhere.
Yes, but only for the mucocutaneous form.
Yeah.
For the cutaneous form, it stays put. You know, you could say this, but if you look at, I know this is not about this subject, but I would just add to the complexity of all this by saying that when U.S. troops went to the Middle East for the first time to fight in Operation Desert Storm, they came back with a form of Leishmania tropica, which is really a form of cutaneous leishmaniasis for the people living in the area. But this form actually visceralized in a strange genetic background host, namely our troops. So they don't always behave the same way in different people.
So by visceralized, you mean it goes to internal organs.
Correct. Whereas anybody living in the Middle East with a particular genetic background, which we don't know about, of course, but we assume it's because of that, the organism stays put in the skin.
Now, in this paper, they say that they call mucocutaneous leishmaniasis. I think we should use the abbreviation MCL.
Sure.
They say MCL development is associated with persistent immune responses.
Right.
Lots of inflammatory mediators like TNF-alpha and various chemokines.
Yep.
T cells coming in. Okay, so
yet they still don't kill the organism.
And they don't kill it, but they stay around a long time. So how long is a typical
This thing can
infection?
last for years.
Years and years?
Yeah, and there's some really horrific pictures of little kids with their entire soft palates eroded away. And you can see right into their pharyngeal area through an area that they can't close because their tissues are all gone.
Now, in this paper, they use a hamster model.
Right.
Leishmania viania
Yep.
is the genus species that they use. And they say the parasites reproduce the metastatic phenotype with primary and secondary lesion development. So that's what you've been talking about.
Yes, that's right.
So I guess you inject the hamster and you get a primary lesion and then secondary lesions.
Correct. And the primary lesion disappears.
Right. So from these hamsters, using the hamster model, they isolated clones of Leishmania that were either metastatic or non-metastatic.
Right.
Right. So some of them would make just the primary lesions and others would go on and spread.
Yeah.
And the initial thing they did was to look at gene expression in macrophages either without leishmania or with either the metastatic or the non-metastatic. And they see that there's a lot of a lot more chemokines in the metastatic, and the macrophages infected with the metastatic variants. Okay?
Yeah, I gather what they set out to do here is understand what the immune system role is in determining whether you get just the cutaneous form or versus the mucocutaneous form.
Right.
Exactly.
So they go into a lot of the immunology of it.
At one point, they start talking about how this is taken into macrophages by phagolysosomes. So, they're phagocytosed, and then the phagosomes fuse with lysosomes and make phagolysosomes.
That's right.
And then they say that there is the innate sensors that are present in macrophages are stimulated, and they wanted to figure out what was stimulating them. And that a good part of the paper is going towards that. And what they find is that it looks like there's a double-stranded RNA, which is stimulating the innate immune system in the macrophage.
My goodness
coming
gracious.
coming from this parasite.
And that stimulation leads to the production of all these cytokines that give the inflammatory response.
Wow.
Right. And so it turns out that the enhanced inflammatory response is caused by the presence of this double-stranded RNA, and it turns out to be a virus.
Unbelievable.
The double-stranded RNA virus of Leishmania.
Wow.
Leishmania RNA virus, LRV1. So they'd have double-stranded RNA genomes, 5.3 kilobases, and they actually, in the cytoplasm of Leishmania, you can see the particles. They're actually icosahedral particles, and there's a very nice drawing over at Viral Zone of these.
So little fleas indeed do have smaller fleas.
That's right. And then they use another set of Leishmania strains that are isogenic, exactly the same except for the presence of the virus or not, and they show that in hamsters. Indeed, when you have the virus present, it metastasizes. And if the virus is not present, it does not metastasize.
Yep. So there are some human Leishmania lookalikes here that you will probably want to look at much more closely if you're interested in pathogenesis. One of them is called Leishmania brasiliensis,
right,
which is the parent strain of Leishmania that causes mucocutaneous leishmaniasis. And the other one is one that's found in the country of Guyana. So they name these strains of parasites or species of parasites after countries. Now, how can that be, Vince? How could you, it's got to be the same species because, just because it's found in Panama rather than in Guyana, it shouldn't be a different species. And yet,
really?
But guess what? Turns out to be the case. They are different species based on their DNA analysis. So there are probably hundreds of different species of Leishmania because it's a zoonotic infection. And it's all over the place. And every time we encroach into the natural world of these parasites, we turn up a whole bunch of new ones.
Do you always see this MCL associated?
No,
or is that unique
not always.
To
No, not always. And in fact, brasiliensis was the only one for many, many years thought to actually do this in humans. And then, of course, they turned up another one, which turned out to be a different species due to the fact that we could now analyze their DNA. Guyanensis is indeed a different species, and it does the same sorts of things.
So you think it's important now to go into people with MCL or just CL and make sure that the virus is in the MCL and not in CL, because this is an animal model.
Right.
It may not recapitulate what happens.
The answer is
in people.
yes. You should probably try to verify this as a pathogenic mechanism in humans.
Now, here's what I don't understand. Maybe someone can explain it. What they say is that, here's their hypothesis. The double-stranded RNA is released from dying parasites early after infection that are killed in the macrophages, and then the double-stranded RNA stimulates the immune system to make all these cytokines. And then they say that subverts the immune response to infection to promote parasite persistence.
I can understand that.
I don't understand why making a lot of cytokines would promote parasite persistence. Tell me why.
Well, you have to know what the mechanism of protection is against leishmaniasis to begin with, and we don't understand that very clearly. But apparently, if you divert the immune system to another pathogen which is present at the same time...
I see. The virus, in other words.
That's right. Then the immune system sort of turns its attention to the wrong thing and starts to try to kill off the viral infection, whereas in reality you want to really zap the protozoan. So these dying protozoans, some of them haven't died yet, and the ones that haven't have now got the option, because the macrophages are busy producing all the antiviral factors.
Okay.
They don't affect the outcome of the Leishmania infection.
Also, I would think by stimulating an inflammatory response, wouldn't you get potentially more macrophages for Leishmania to infect?
You actually,
yeah, good point.
you're attracting more host cells to the
Okay.
area too. Exactly right, and more carriers.
And you're mobilizing them to other parts of the body, which
Here,
is
here.
going to carry the parasite to the next mucosal
So you have
layer.
a double whammy in terms of that.
So that explains
See,
the
I
metastatic
like-
part.
They didn't really say that, but I like that better from the virus point of view. That suggests that this actually could be a virus-directed mechanism for its survival.
Yeah.
It's
Because
helping its host replicate.
What's interesting here is that these viruses are not extracellular, as far as I know.
No,
They're just passed on by cell division.
that's right.
And even though they're encapsulated, which to me is weird to have a capsid, they don't get passed outside of the cell. So, if you were going to persist, if the virus wants to persist, this is a good strategy for doing that.
Yep.
Well, at some point in its evolutionary history, it presumably needed that capsid.
It might still need it. Like TY elements have capsids, and the capsid structure is probably part of the polymerase mechanism. Right? It keeps the nucleic acid and the polymerase in one place.
Yes.
I guess it's also
Yes.
possible that it needs it in the normal animal host, right?
Yeah. But I mean, if you imagined it without a capsid, then the polymerase and the genome, if
It
they
could
float apart, the stoichiometry gets.
Yeah, because these double-stranded RNA viruses, they need to have polymerases in the capsid, right?
Yeah.
So that's
Right.
a good point. Now, why in nature are there... So we're going to jump to nature now. We're not sure about this. So why in nature would there be some strains with and without this virus? It's just the way they
Well, no,
they
wait a minute. You
originated.
can say the same thing about all these viral infections of bacteria as well. For instance, with cholera, you've only got one species of cholera that actually has a prophage, and all the others don't.
Okay.
So it's just a matter of chance and luck.
It's a luck of the draw, right?
It's the luck of the draw.
Okay.
Right, and this virus happens to give it possibly gives it a selective advantage in this new environment in humans.
Yeah.
So eventually, would you say that all the Leishmania is going to have this virus then?
Depends on which animal species gets infected and gets crossed with others. I mean, this is a zoonotic infection, basically.
So the reservoirs are animals?
Yeah.
What animal is it again?
All of them.
All of them.
If you want to know which animals in South America harbor another pathogen not related to this one exactly, but it's in the same group of Mastigophora, the flagellated protozoans. You look at Chagas disease, for instance, which is also an amastigote-like infection in the tissues, at least. Every single mammal of South America has it. And by the way, you'd think of Africa as the most mammal-rich continent, but actually it's South America. There are many more species of mammals living in South America than there are in Africa.
Okay.
And the reason for that is that there's less predation among the South American animals than there is
Right.
among the African animals. And so it limits the number. At any rate, you might see a spread to them, but it's such a complex web of intrigue in terms of an ecological setting that... And you have these little microzones throughout the Brazilian jungle that they'll never get together, basically.
Okay. Perfect sense. Dickson, we can call this a parasite of a parasite, right?
Absolutely.
Fascinating.
You betcha.
Oh, you
That's great stuff.
All right.
There are no clear-cut lines between pathogens and hosts.
Apparently not.
None.
Our next paper is dealing with a new virophage. We talked about virophages on TWiV Infects Fib, if you remember, Dickson.
I do
Yes.
remember.
That was the second known virophage. The first is Sputnik. So they're called virophages because they infect only cells infected with another virus, viruses so far, and they inhibit the production of those viruses. So that's why
Right.
viro-eater, because phage means to eat,
Right.
right?
Yep.
Not sure that the perfect name, but I think it will stick. So this study, which is published in PNAS, is called Virophage Control of Antarctic Algal Host Virus Dynamics. So basically they go to an Antarctic lake called Organic Lake, And Dickson, have you been there?
No, I haven't been that far south.
At which lake were you telling me about?
Lake Vostok.
We have an email about that today.
Yeah?
You're going to like it.
Lake Vostok is a lake that's actually below the ice sheet of Antarctica, about a mile below the ice sheet. How can there be a lake down there? And the answer is that there's a vent that's hot that keeps the ice melted underneath the ice sheet, and it creates this liquid zone, and the zone is about the size of Lake Ontario.
Yeah, so we have a curious email. Which
and the Russians are busy trying to drill into
exactly.
it.
That's what it's
and
about.
They haven't quite got there yet, and they're afraid to contaminate it because they're not sure of what to do next. They'd love to know what swims in it, though.
So this is a surface lake, and what they did was they did metagenomic analysis of the surface water of organic lake. So they took water and they filtered it through 3 different size filters, 3 different pore sizes, and they sequenced it. And they basically found... So this lake has a lot of... it has microbial communities, and it's dominated by algae.
And it's very salty. It's not
Extremely
a freshwater
salty.
lake.
Yes, that's right. Hypersaline lake. And they found a number of different phages. These are large DNA viruses that infect algae. All right? They call them organic lake phages. Among the sequences, they also found sequences homologous to virophages. Now there are 2, so you can start comparing whatever sequences you get to the database. There's another one, and so all of this paper is really sequence analysis. There's no experiment here, which is okay.
Yep.
They say, okay, we've got another virophage, and this virophage looks like it would likely infect these phages. So they call this the organic lake virophage.
So you'd think these little viruses are protecting the algae from destruction for the viruses.
That's what their theory is, yes. So there's a lot of assumptions here. First of all, the assumption is that this is a virophage of the phages in the lake. And the assumption is also, like other virophages, that it inhibits the production of that virus.
In a mammalian cell, interferon is a primary defense mechanism of host cells, eukaryotes. Well, an algae is a eukaryote cell, but it's a plant.
So you're saying that a virophage is...
do plant cells produce interferon?
I don't believe they do, no.
Or an interferon-like molecule?
I'm quite sure they don't make it... No, maybe something that's analogous functionally, but they don't make it.
I mean, how do they protect themselves against viral infections, then?
With virophages.
Maybe virophages. Maybe they remain to be... Now that we have sequences, people will start discovering them. So the assumption is that these are protecting the algae, right, from the phages.
That's
and
wild.
So they do a modeling study. Basically, it's called a Volterra model. So I have never encountered this before. Do you know about that?
Sure, of course.
What does it do, Dickson?
Well, these are 2 ecologists that studied. Believe it or not, there are two ciliated protozoans. Mm-hmm. One is named Didinium, and the other one's named Paramecium. And Didinium eats Paramecium. So if you have a clear aqueous solution, and you throw Paramecium in first, and then you throw Didinium in, eventually Didinium will eat all the Paramecium. Mm-hmm. But if you throw in a layer of muddy water at the bottom and allow Paramecium a refuge so that Didinium can't see, in quotes, the Paramecium seems to survive quite nicely down into this... And Lotka and this other guy spent their whole lives, Volterra, studying the dynamics of predator-prey relationships based on their observations of Paramecium and Didinium. So they're very interesting people.
Here's why I don't understand this, because it's based on a first-order nonlinear differential equation. So that's
exactly
it for me.
No, I never said I understood it. I do know who those guys are, though.
So these models describe the oscillation of predator and prey.
Yeah.
So here they apply it to this lake, which is, as I said, mainly microbial. There's very little multicellular eukaryotes here. Yeah. And they say the virus preys on the algae, so they model the cycles of algal blooms, and going away, then the virus rises. And they say basically if you throw the virophage into this equation and you assume it inhibits virus production by 70%, which is what Sputnik does to virus, then you get more cycles of algal blooms as a consequence.
And you also get less total mortality of the algae.
Exactly.
Absolutely.
So more of them survive each boom, and then they can restart the next boom faster.
Yeah.
And this
it's important to keep clear that this was modeling now.
Yeah.
Yes.
Yeah, exactly. But there's some real ecological studies that back these guys up. There's one up in Grand Isle, up in Lake Superior, that's got just two things on it: moose and wolves.
Yeah, and they can apply this equation to it.
Yeah, they can, because the wolves will eat themselves out of house and home, and there are very few moose left, and there are lots of wolves, and all of a sudden there's a crash in the wolf population, and then the moose population goes back up again.
That's great.
And it just keeps fluctuating back and forth. Owls and voles are the same, with lynx thrown in just for good luck.
So I don't know this field, going into lakes and doing this field stuff, but could you go and now sample this lake periodically and confirm this?
Well, they didn't they have a subsequent sample taken a couple of years later, and they confirmed that the virophage was still there?
That's right.
Yeah.
And that it had not changed significantly.
It had not changed much. But the question is, could you stay there for a year and sample, every day and do this?
Right. You could do it, but it would be wrong.
I don't know if these guys do this or if they just model things and make conclusions.
Well, no, they take
that.
small samples and they expand out to the big sample, I think.
Well, I think if you're volunteering to sit next to an Antarctic lake for a year,
forget about it.
They'd probably be very interested.
I think all year long it's not a lake. Sometimes it's completely frozen.
You know what I couldn't help think about, and I might have the details wrong here, but this what we used to learn about hepatitis delta virus, which was the parasite of HBV, and it could be more complicated like that, right? Because there you had a parasite of a parasite, but the result was not better for the actual host. I think it made the HBV infection more severe.
So like a helper virus rather than
that was the original idea, but I think those are not. That's not long.
By the way, it should be mentioned in passing, speaking of hepatitis, that Brooke Blumberg passed away just day before yesterday.
That's right, the discoverer of Hep B, right?
Yep.
Yeah, Welkin, my understanding is that it's no longer believed that Hep Delta co-infection with Hep B makes it more serious. I don't follow the area, but I've just heard that recently. So I would go sit by, if this was my shtick, I'd go sit by the lake as long as I had a little cabin that I could sit in. And if I had high-speed internet.
Right.
And an iPod.
That's right.
In my TWiV studio, I could do podcasts,
I think what I would do is see if I can
How's the weather there today, then?
Right.
That's right. What weather? I can't see out my window.
Sorry, Alan.
Right. What I would do that wouldn't involve sitting next to an Antarctic lake for a year is try and get a strain of these algae cured of or natively uninfected with the virus and the virophage, and see if you can reassemble the system in the lab.
I guess,
that's right.
Could try and do that, sure.
Because the other thing that occurs to me with the lake year round is not only is it going to freeze over pretty solidly in the winter, it's also going to be really dark. And so
that's right.
photosynthesizing algae, you expect them to just kind of taper off.
And in fact, that's one of the points made in this paper, that when you don't have a lot of light, maybe these virophages are part of the adaptation so that these blooms can go more rapidly and to higher extents. And then you when it's dark, you don't have to worry as much. You understand what I'm saying? That's the idea.
Yeah.
It's
so how did an alga that's in the Antarctic lake in this isolated place that's infected with a virus, how did it pick up a virophage that's so useful to it?
Came from the air.
Came from the air.
It could have, actually.
Sure.
It might have. Is it air currents?
But you don't know how old this relationship is either, because Antarctica at one point was tropical.
Right. And also, actually, I guess in this paper they looked at samples from the Galapagos as well.
That's a good point. They actually have looked at a number of different environments.
And even exotic places like Delaware Bay.
And they're finding them?
Yeah, I thought you would like that, Alan, Delaware Bay.
Yeah, I appreciated that. It's one of the worst places in the world to sail a boat, but apparently it's a good place to find.
It's like
they found virophages there. They found them in a fresh and hypersaline tropical lake, a neighboring lake in Antarctica called Ace Lake, and an estuary. So they think maybe they're in a lot of different water systems, and they could. So their conclusion is they may influence overall carbon flux, in these different aquatic environments.
How many different species? How many different kinds of viruses are there?
So now we have 3. We have the original, and then the original was Sputnik, and then the one we talked about last week was also of one of these big viruses, the cafeteria renbergensis virus.
Oh, yeah, that's right.
Which was an amoeba, right?
Yeah.
And now we have one of a different kind of host, a virus that infects algae. So I would like to know if there are virophages of mammalian viruses, right? Why not?
And I'd like to know if anybody has are there any Mimi-like viruses in that size class that don't have virophages in them?
Yeah.
Remember that saying, though, successful systems attract parasites.
Yes.
Maybe it's a big virus that attracts a virophage. That's a good point. Maybe the smaller ones like pox virus. Rich is not here. He can't.
Maybe they were derived from the big virus.
It could be. In fact, is there some evidence of lateral gene transfer here? I think I seem to remember
sequence between the small virus and the big virus.
I remember the cafeteria virophage had, DNA transposon homology. So
yeah, actually, I think in this paper they did find
there was a set of genes.
Yeah, there's a set of genes that appears to have come from the virus that it's parasitizing.
Isn't that ironic?
Yep, that's right. Gene exchange.
No, just resourceful.
Between the virus and the virophage. That's
right.
No,
it just means that evolution is a never-ending series of mirrors that are facing each other.
You're right. It could have come from the virus. Yeah. It could have been selected as a way of regulating the viral killing.
So, gentlemen, here's a question for all of you, because Baruch Blumberg, I did mention him. He was involved in exobiology for a while for NASA.
What's that, Dickson?
Exobiology is the hypothesis that suggests that there's life somewhere else besides on Earth.
Was that arsenic-based?
I don't know what it is. No, but the question arises then, if life exists somewhere else, is it parasitic life also? Do you have parasites of that life, of those life forms as well? Would you expect that if things followed along a typical path?
I would expect that.
I would too. Absolutely.
Yeah, given that just about everything we look at, that every species we look at has parasites. This is something that developed very, very early on in evolution.
Sure. Now remember, this is our word for those life forms. We call them parasites. But in nature, they may be regulators. Yeah. Okay, they're actually life regulators.
Yeah, we give our own names.
Without parasites, I think this place would be a mess.
Something like this, with the algal blooms, you've got it not only regulating the algae process, but possibly the global ecosystem.
Hear, hear.
I mean, they seem to find this in water all over the place, and algae are a huge part of the carbon cycle, the nitrogen cycle, the oxygen cycle.
You betcha.
So to what extent does life on Earth owe its existence to virophages?
Sure. Or just viruses in general.
Well, we've certainly appreciated in recent years how much we owe to bacteria in archaea.
Yeah.
And maybe we will learn about how viruses contribute, and then virophages.
Hear, hear.
Has anyone asked whether or not the genes in the virophage that are borrowed from the host virus, whether that's whether they're simply ripped off and being used, or if it indicates some sort of dependency? Like mitochondrial DNA carries genes that determine compatibility with the host cell. Is it possible that it's a system like that?
Right. And how different are they actually from the parent organism? Have they evolved on their own after they split?
They have, yeah.
Well, there you are.
That's a good question. I don't know the answer. Let's see. There are 6 genes that they identified in the virophage that have here's the identity: 6 proteins, 32 to 65% identity to sequences in the virus.
So they've been around a while.
Yeah, it's a lot of evolution, right?
I mean, these lakes have probably, I assume, I'm going to assume here, but I shouldn't, but in Antarctica, it's got such an unusual geologic setting that it's hard to imagine things being permanent there, except for the ice sheets. But if you look back through history, you can find fossils of fern plants and dinosaurs and all kinds of other things. So at one point, this was a verdant tropical zone.
So Welkin, they say that, they don't know what these proteins do, but one of them, they say, may mediate helper virophage interaction. Other genes are transmembrane protein, phage tail fibers.
Wow.
collagen-like proteins, and
All
they
kinds of
are
stuff.
really on this idea that they may facilitate interactions between the two. So it's a good question. One thing I didn't appreciate up until now is that these virophages have circular double-stranded DNA genomes. For some reason that hadn't come up previously. And there's a nice picture of them in this
Makes
paper.
their replication cycle a little bit more complex.
I think it's that
ends.
picture that made me think of mitochondrial DNA
Uh-huh.
or
Yeah.
chloroplast DNA.
Uh-huh.
Yep.
You know who must be going nuts over all this is Lynn Margulis.
Yeah, yeah.
Hey, you know, Coulter mentioned the Gaia hypothesis the other day.
Good for him.
Because he was talking about these these little algae in the
Coccolithic phytoplankton.
yeah, in the English Channel that mediate
sure.
the weather.
Absolutely.
It's
Yeah.
amazing.
They make
Amazing.
clouds for
Yeah,
him.
exactly.
No, I want to talk to him.
Welkin, have you ever seen these coccolithic
Phytoplankton.
phytoplankton?
No,
They have
You'd love them.
their covers are.
I've never
really-
met that man.
They're really regular.
The next time you take one of these guests out for dinner, introduce them.
The covers, the shells on these algae are very regular. They almost look viral in origin.
Well, they're made out of calcium carbonate.
yeah.
So they sequester carbon, and then when they multiply enough in the ocean, they actually sink to the bottom of the ocean, and then they recycle carbon.
So that's what makes up the White Cliffs of Dover, I understand. That's what
quite.
Coulter said.
No, it's more complicated than that.
Yeah, he said this is all the dead coccolithophores.
Well, there are some in there. There's tons of them, no question about it.
But
But there's
other
other
stuff,
things.
too.
So yeah, there's
All
other
right.
things.
Okay, let's move to our third paper.
You're right. I am sorry I missed his talk.
I didn't tell you had to apologize, Dixon. It's okay.
That's okay. You can bleep it out if you want.
You were doing important vertical things.
Yeah, exactly. because soon we shall all be horizontal.
Well, we are anyway. It happens. The third one is in emerging infectious diseases. It's called human metapneumovirus infection in wild mountain gorillas of Rwanda. And this is from Gustavo Palacios, who was on TWiV a long time ago.
Yes.
He's from the Center for Infection and Immunity here at Columbia, headed by Ian Lipkin.
Who is the senior author on the paper?
He is. And this one, Dickson, you may know about. They're talking about the world's remaining gorillas.
Yeah.
Mountain gorillas, right?
Mountain.
So there are other kinds?
786 of them.
There are highland gorillas and there are lowland gorillas, and in fact, there's a ton of lowland gorillas. But
So the mountains are the highland gorillas, right?
absolutely.
So the lowlands, there's much more.
Yeah, they just discovered.
And then there are the gorilla blends, which compare characteristics of both.
That's right. And the decaf and the
There's a nice glue called Gorilla Glue.
No. They discovered imagine this now. How unlikely is this? About 2 years ago, they announced the discovery of a colony of lowland gorillas, 125,000 gorillas.
Where's that?
I believe it was in Rwanda, but
Oh.
I'm not sure about it. But how could you overlook 125,000 lowland gorillas? But they did.
Well, in this paper, what happened? These gorillas apparently live in parks, right?
Well, they've been set aside for them because they
You
realize
can't,
they're so rare.
But they're not zoos, right?
No,
They're-
of course
They're
not.
open, but you can't build in these areas.
Well, they're
so
wildlife preserves.
And then there's a lot of ecotourism, right? You can go.
Correct.
What do you do, Dixon? You go and you watch them?
You do. You sit there and you're not supposed to make eye contact.
Is that right?
Yeah.
And you can get relatively close to these
You can
gorillas?
have them pet you sometimes.
Oh, I
You
didn't
can
know
reach
you
out.
could touch them.
You can look away and reach out with your right hand, and they'll reach over and touch it.
And why can't you make eye contact?
Because then they think it's an act of aggression.
And they would kill you.
Well, they could. They could very easily kill you. They're very strong animals.
Yeah, they are.
I mean, they can rip a tire apart. All right? They can take their bare hands and reach inside of a tire and break it apart. That's pretty strong. That's a steel-belted radial tire, too.
In fact, it says here, the parks are surrounded by the densest human population in continental Africa.
Yeah,
75% of these mountain gorillas are habituated to the presence of humans.
that's true. And they poach them. Unfortunately,
Really?
they kill them and they sell
I heard they
their
were better
parts.
fried.
Stop it. Just cut it out.
So there was an outbreak of respiratory illness. Actually, there apparently have been sequential outbreaks.
Well, they were
4
susceptible to
different groups.
Ebola for a while, too, as I recall.
Yeah, I remember that Ebola is decimating. No, not decimating.
No.
Sorry, Dixon.
That's okay.
It's reducing the numbers,
That's
right?
correct. That is correct.
So there were some outbreaks, and that's what this paper has done. They went and obtained samples
Yep.
these gorillas were having coughing, nasal discharge, lethargy.
Yep.
And in fact, one group which they studied, called the Hirwa group, 12 animals, 1 adult male, 6 females, 3 juveniles, and 2 infants. So that's typical, just 1 male.
Yeah. For them it is.
They got moderate to severe disease in 11 of the 12 animals.
Yeah.
They don't know how they got it. In the end, they're not sure.
Yeah.
And a few of them died. 2 untreated animals died, an adult female and 1 male infant.
Yep.
And so they have a lot of descriptions of the clinical symptoms.
Do they think the virus came from humans?
Yes. So they
Yes.
so they did deep sequencing.
All right.
Actually, they did multiplex PCR initially for respiratory pathogens.
Got it.
So you can use primer sets that would multiply everything you suspect to be infecting the respiratory tract. And they found human metapneumovirus in serum, lung tissue, throat, nose, anus, and vagina swabs from the adult and in lung tissue from the infant.
Oh.
They also found streptococci and Klebsiella pneumoniae from the adult gorilla.
So this could also be a TWiV.
It could be. That's right,
Absolutely.
because apparently the metapneumovirus can make you more susceptible to pneumonia,
Right.
bacterial pneumonia. So they say, well, the fact that we find this virus suggests that it infected them. And of course, they can't prove it. But in one of the animals, it was the sole pathogen. The infant
Right.
supports the
Yeah,
presence
and
of
they
this agent.
actually, in terms of the source of it, they actually traced the particular sequence of this metapneumovirus, and it matches a human a strain from South Africa.
Right.
This is a big worry. I mean, it's not just among gorillas. I mean, anthropologists worry about this happening all the time when they discover new naturalistic peoples living in, let's say, the Brazilian jungle, for instance. They want very few contacts between themselves and these people until they work out who they are and what diseases they've already encountered.
Didn't you once say that you thought this was how Homo sapiens wiped out the Neanderthals?
You bet I did, and I think that's absolutely right because the wipeout happens so fast, and we have genetic remnants of the fact that we weren't exactly hostile towards them because we have a portion of their genome in ours now.
Yeah.
So the fact is that we probably killed them off with microbes inadvertently.
Well, anyway, if you want to know about metapneumoviruses, they are a relatively recently discovered member of the paramyxovirus family. Same one that would have measles in it and mumps and other respiratory pathogens, the para-influenza viruses. They were discovered by metagenomic analysis of samples from people with respiratory disease.
Interesting.
So they account for a good amount of human respiratory infections. So they're common in humans, so it's not surprising that they would jump over to an animal such as a gorilla.
Right.
Or any other animal. I don't know how close you would have to be to get infected. It's an interesting question.
That's our closest relative.
The
in terms of-
gorillas?
the upland gorilla.
I thought
think it is.
chimpanzees were.
No, I think the gorilla is.
Welkin, you ought to.
Actually, yeah,
Come on, what is it?
They're both very. I think chimpanzee wins the tie,
Really?
but gorilla's very close. So it was controversial for decades.
Well, I'm more bonobo-like in
but-
terms of that.
But it actually, reminded me that actually this same thing happened in a chimpanzee too, right? I think Vince blogged about a case in a zoo
That's right.
where the same virus, I think it was a metapneumovirus, got into zoo chimps.
Is that right? It was in Let's check. We'll look
I
that
think
up.
so.
It was in the Lincoln Park Zoo. I think that's
That sounds
Chicago.
right. Yeah.
So
that
primate
Chicago?
colonies have big worries in terms
You're
of that,
absolutely right. Human metapneumovirus. A 9-year-old chimpanzee died of respiratory disease caused by a human metapneumo, and that was in a zoo. So that virus, here's the number I was looking for. It's responsible for 10% of all human respiratory tract infections.
yes? Do we die from it?
You can get
Apparently, I gather the secondary infections are, in some cases, the bigger
Okay.
risk
Yeah.
than the
Okay.
That's right.
virus.
You get secondary bacterial infections.
That
But these
makes
were that was a chimp. And then there have been infections of bonobos in wildlife sanctuaries by influenza.
So, Welkin, how far apart are chimpanzees from bonobos?
They're. I don't know off the top of my head, but they're probably like Homo sapiens and then Neanderthals. I don't think
okay.
they're that 1 million or 2 million years. I don't remember.
I mean, they're quite different, though, aren't they, in terms of their demeanor?
That's my understanding. Is the bonobos are the ones that actually have a lot of human-like behavioral characteristics.
Yes.
So, Welkin, you had raised this interesting question about how phylogenetically, how far apart.
Yeah, this is one of the points that Eddie Holmes makes all the time. He sort of has a list of fundamental questions that we don't understand yet about the principles by which viruses jump between species. For example, are certain types of virus more likely to do this than others? When a virus does this, does it have to go through a period where it adapts to the new host? Did these gorillas get it directly from humans, or did it have to spread in the gorillas for a little while? And then sort of the big question then is, are viruses more likely to succeed in a jump if they're jumping between two closely related species? And I think instinctively a lot of virologists immediately they think of counterexamples, because the things we know about are viruses from mosquitoes or viruses from rats or viruses.
Well, and also
bats.
because virologists are smartasses.
Yeah. I know two or three like that. They like to podcast too.
No.
Sorry. But there could be some sampling bias here too, right? Because many humans, including the ones that live in countries with medical surveillance and things, are not living near other primates. So the real way to answer this question would be to go and look. And some people have done this. If you go look at humans that live in regions where there's primates with endemic viruses, you do find that humans are often seropositive for viruses of monkeys and chimpanzees and things like that.
Now, the term, when you define success of a jump, you mean a virus that not only jumps from an animal to a human host, but actually is then able to sustain itself human to human?
Yeah, I guess success would be manages to get a foothold and start to spread.
I mean, like SIV to HIV, and then after that.
So
right. So if you look at something rabies going from a bat to a human, it's not a successful jump.
Well,
no.
Not when it's 100% fatal. It's not, Alan.
Depends who you ask.
There's no chance for it to take hold.
The SIV is an interesting example, because we have the SIV from chimps that gave rise to HIV-1, and that has spread all over the world. But there's also the SIV from pseudomangabes, which is not as close to humans as chimps, and that's HIV-2. And it's interesting that the HIV from a more distant monkey has not spread well in humans. It's it's tended to stay in the regions where it first made the jump.
But Welkin, if it's correct that SIV went into people around 1920 or so, but it took 60 years almost for it to really explode. And is that a period of adaptation, or?
I think it probably is.
Wow.
I don't. I can think of one piece of evidence that hints at that, and it's Beatrice Hahn and Paul Sharp have I can't remember where the paper is published, but they did a big phylogenetic analysis, and they sort of they took alignments of all the SIVs and HIVs, and there's certain amino acid positions that are always one thing in humans and a different thing in non-human primates. So there's one amino acid in matrix that always changes when a virus jumps into humans, whether it's HIV-1 or HIV-2. And it's even the case that experimentally, when someone put HIV-1 back into chimps in the 1980s, that amino acid flipped back to the SIV version.
Right.
So nobody knows why that is, but there's, I think, examples like that tell us that there must be some adaptation to human genetics going on.
Fascinating.
Interesting.
Yep.
Interesting questions.
Yeah, and I guess it's not just a virology question. You can apply this to any zoonotic transmission.
Sure.
Does it make it more likely if it's more closely related?
Sure. It depends on how far back you want to go, but if you go back more than 200,000 years, they were all
right, they were all
zoonotic.
non-human, yes.
That's right. So is that a long period of evolutionary history, or is that a short period of evolutionary history, 200,000 years? It depends on what you're talking about, I guess, and what the selective pressures are for each one of those things.
It's a long time for virus.
It is, but I mean, if you look at the evolution of, let's say, salmonids, the trout and the salmon family, it's only been around for 20,000 years. You've got all these species of trout and salmon out there, and before that, they weren't there at all. So that's remarkable. Depends on the selective pressures and the reproductive rates and stuff like this, I guess.
All right.
Anyway, the current cover of Science magazine has a picture of a salmon on it, by the way.
Why is that?
Well, because they were looking at different strains of these sockeye salmon with regards to their temperature tolerances, and in a very short period of time, they can adapt to a new environmental change.
Meaning how short?
20, 40, 50 years, that sort of thing, which is very short indeed.
I think for us it's short. For a virus, it's very long. Especially for phages, which replicate in 20 minutes, right?
Yeah.
Let's do some email.
Okay.
The first one is from John, and he writes, "It seems the Russians are going to penetrate the oldest subglacial lake on Antarctica soon." He sends us a link to a New Scientist article, which says, "Mysteries of Lake Vostok on the brink of discovery."
Yep.
So John wants to know, I really don't care if they find oil or natural gas there, but I would be very interested to know what they find if they get a nice microbial soup they can sequence. I'd be especially interested in adding to a timeline of viral DNA that's inhabited other species' genomes. So the question is, from a microbial perspective, what would we expect to find there, and will we get any cool stories about it in the next few years?
Wow. Well, that's a great question.
Yeah.
There will be microbes there, right?
One would assume so, because of the events of warm material coming up to keep it in liquid form. It's been there for millions of years, too, so that's very interesting. It's been sealed off for millions of years, but it's been a liquid lake for millions of years. So the answer is you should find lots of interesting things in it.
Do we know geologically if it was a liquid lake when it was sealed off?
We don't have any clue about that.
This sounds like a Geraldo episode. do you remember when Geraldo opened Jimmy Hoffa's safe?
Yes.
There was nothing in it.
Yeah, or the safe from the Titanic.
Wasn't it Al Capone's safe?
Or
Al Capone. Well, big deal, same deal, right? Different spelling.
This won't be empty, I'm sure.
No, Lake Vostok will have some things in it. It could have a rich protozoan community also, so you don't know.
But anything here doesn't need light, right?
Right.
It would be interesting if it's something that we've never seen a homologue of.
Yeah, it's a mild...
Something with no relationship to anything else.
Yeah, but I don't think we'll see that.
You know, it's hard for me to imagine is a mile thick worth of ice. I mean, that's just...
Yeah.
I see a sci-fi movie in the office.
Look, I think it's going to be a subset of what we already have found.
Well, the thing is that the probe itself may contaminate the lake. Even if there's nothing in it, eventually there will be something in it.
Yes.
Just like the probes on other planets that we put there could have seeded those places with life forms.
So how thick is the ice above the lake, Dickson?
It's a mile.
How are they going to get through?
They're doing it through this...
drill, baby, drill.
It's a drill with a heated end to it that sort of melts its way down through.
So a mile is not a long way to drill, right?
No, not in terms of oil people.
Oil people drill more than a mile, right?
Oh, absolutely.
Right? Okay.
Oh, there's a gold mine in South Africa that's 5 miles deep, so...
and mine drilling technology, what could go wrong with that?
Yeah, exactly. You're right.
That's my point precisely. You said Geraldo? Okay, fine. You put on Oprah also.
I read this article. They say the Russians aren't worried because the water is going to shoot out, so they're not worried about contaminating it.
You think because of the pressure?
Then they say it will refreeze, so it'll be fine.
Oh,
so
it's interesting. They say that they'll let it refreeze and then go back and sample it a year later.
Yeah.
Did you see that?
Yeah.
So they're going to drill a hole, let some water come up into it?
Yeah. Sample the water, let it freeze, and then come back later, yeah.
Very interesting indeed.
Well, if they find E. coli 0157, they'll know they contaminated.
That's right. Or they look down on the bottom and they see an empty newspaper laying open. You know?
All right, sorry, go ahead.
Vince's original poliovirus clone is in there.
That was oh boy.
That's right.
I didn't send it to him. He continues, one final note, I am an economist by training, and in the fall of last year, the NSF sent out a call for white papers to get the opinions of the field on where research is or should be headed in the next decade in all subfields of economics. I haven't had time to sift through all of them, but I thought it would be an interesting thing to touch on TWiV. e.g. long-term ongoing studies, novel ways of using viruses as a tool, either natural or engineered, etc. Yeah, we could do a whole show on that, right? Dixon?
Sure. Absolutely.
That's a good idea. I'm going to put it in the future show.
Yeah.
So you mean like predictions of where virology will go?
Yeah.
What will be figured out? Yeah.
Oh, we've talked about them being used as batteries and stuff like this. Why shouldn't we expand the field?
You could do that, Welkin, right?
Oh, yeah.
Thanks again for the entertaining education and a belated Happy New Year to the crew. He says, P.S. Dickson, another TWiP is spiral. Is clearly a masterpiece tagline and should be reconsidered. I know you don't like it, Dickson.
That's all right.
Sounds like a friend of Alan's.
Yeah, exactly. They party too much together.
The next one is from Daniel, Dr. Daniel, PhD. Dear Vincent, I've been listening to TWiP for the past few months and then started crossing over to TWiV and find both podcasts refreshingly spontaneous. I download these and other science-related podcasts and play them on my iPod when I take my morning walks along the Clearwater River Canyon in northern Idaho.
I know where that is.
You do?
I do. They have remarkable runs of steelhead trout up that river.
Mm.
He writes, I'm a lipid biochemist currently working on neuropsychiatric disorders. Specifically, I'm examining the methylation patterns of certain neurochemical and peptide receptor and neuroimmune ligand receptor pairs in rodent brain from animals reared in different social and stress-inducing environments.
Wow.
I also teach a medical oral micro course in the dental school. I'm looking... This guy sounds like you, Dickson.
Wait, which school?
University of Washington State University and Eastern Washington State University.
At Pullman.
Is that in Pullman?
Yeah.
I don't know where he's walking in northern Idaho then.
Well, it's not too far away from it. I guess he must take his car and go.
Maybe I got it
to
wrong.
Clearwater.
His abbreviation, Clearwater River Canyon in NO.ID. Wouldn't that be
Idaho?
Northern Idaho? Okay.
Idaho.
I'm looking into the molecular genetics of inherited and somatic environmentally acquired CpG methylation and the potential mechanisms underlying these covalent and reversible modifications. After listening to your podcast in both TWiP and TWiV, I am obtaining more discrete information about the tremendous impact that eukaryotic parasites and certain viruses have on both the architecture of a mammalian genome and the activity of the immune response. I believe these are co-adaptive phenomenon. My question is directed to you in both TWiP and TWiV co-hosts. What do you know, and could you speculate about the impact of parasitic challenge and or viral infection on the early development of the immune system as these relate to the accumulation of copy number variations in the human genome?
Well, I can answer the location question you were just discussing anyway.
Please do.
What's that?
He's at Washington State University in Pullman, Washington.
Yes.
Which is just west of the Idaho border.
Oh, okay,
Correct.
so he can walk there in the morning.
I don't know.
Well, I don't think so. It's probably about 20 or 30 miles away.
5 miles to the
Is it
Idaho
5
border.
miles?
I
That's
don't know
all.
how far it is to the
To the Clearwater.
Okay.
to the Clearwater.
Yeah, well, tell him if he needs somebody to help him walk along the river, I'll be glad to join him.
I'm sure you would. All right, what about copy number variations selected by parasitic or viral challenge?
Interesting.
So I think I know what he's asking, or I could
Go ahead.
kick this off. So copy number variation, in the specific sense, is a term that's actually just cropped up about 5 or 6 years ago. It's kind of surprising, but it was only in 2004 that geneticists figured out that actually a major proportion of polymorphism between people is actually copy number variation. It's where whole segments of chromosome are expand and contract in copy numbers, so that even closely related people might have different numbers of the same gene. So some individuals might have 4 or 5, and others will have 1 or 2. And I think up until 2004, people thought that must be rare. They assumed it would have such a massive phenotypic consequence, like trisomy X or something like that. That they assumed it was rare, and it's actually turning out to be probably contributes just as much as nucleotide polymorphism to variation. But what's interesting about copy number variation, if you have more copies of genes, in theory, you could have things like more expression of a particular protein, right? If you had 3 genes expressing the same protein.
Right.
And so I think maybe what he's asking is whether parasites and viruses have sort of driven or maybe select for copy number differences.
Right, that's correct.
Or the maintenance of polymorphism in immune system genes.
Wow.
And the MHC is a great example, right? If the MHC has been around a long time, but if you look between different primates, there's huge differences in copy number, like whole sort of meta loci that have been duplicated, expanded, and contracted.
If he's asking on the individual scale, though, that might also, I guess that might also happen where somebody who's exposed to viral or parasitic infection early in development might develop more copy number variation than somebody who wasn't exposed?
That's the thing, because he started off talking about epigenetics in the paragraph before, so then I thought maybe he's asking that, and it is true that unlike nucleotide polymorphism, which is the result of a very low mutation rate of polymerases, copy number variation arises by recombinogenic mechanisms, and those go on all the time. I think I've probably even heard that like identical twins will have copy number variation.
Yeah, I was going to suggest looking at triplets or identical twins to see whether or not that's actually the case.
So do pathogens
he might be asking that, yeah.
Yeah, so do pathogens leave scars on our genomes?
Right.
I mean, I suppose there's proof in principle there with antibodies, because antibody genes, individual B cells,
sure,
generate new antibody combinations essentially by a recombinogenic mechanism.
Mm-hmm.
And then those particular B cells will get selected if they're specific for something, those will expand. So maybe, I guess the answer is I think there's a good chance that that's true.
Yep.
I found a very nice article in PLOS Genetics, copy number variation of CCL3L and the coevolution of primate and viral genomes.
Yeah.
And this is actually an AIDS, HIV, SIV paper. You probably know some of these authors, Guido Silvestri.
Yeah, this is actually turning out to be a controversial.
Yeah.
And it's an interesting. There's two parallel stories going on. So the original publication a couple of years ago claimed that copy number variation in this gene that expresses a chemokine that happens to be a chemokine that binds to the HIV receptor could affect whether or not individuals are susceptible to HIV-1. So if you made more of the chemokine and it was blocking HIV-1 binding, maybe you would be less susceptible to HIV-1 infection. And so the original association study in human cohorts suggested that was true. And then there was a parallel study, and I think it's mentioned in that article, in SIV-infected macaques claiming the same thing.
Mm-hmm. Right.
The problem is that a couple of other groups got into this, and so there's groups at NCI and in Chicago that have tried to reproduce the results in human cohorts, and they say they don't see it. And Norm Letvin's group has tried to reproduce the results in the monkey experiments, and they don't see it. So there's 4 or 5 different groups involved working in 2 different one human system, one animal system, and so it's interesting. But I think the verdict is still out on whether or not it's true.
Well, we'll put a link to this in the show notes. It's open source, no, not open source, open access, and there are actually some references to some other articles on the same topic for Daniel, so you can check that out. Sorry for this long-winded email, but I think some background was necessary to frame my question coherently. P.S. On an unrelated topic, what does Dick think about the similarities between nurse cells and the Rhizobium species root nodule involved in symbiotic nitrogen fixation in leguminous plants?
Right. Well, there are some parallels in terms of biological nurturing, but as far as how the structures actually form and their...
What is a nurse cell?
Oh, well...
I know I learned that.
No, that's okay. Welkin, it was one of my first TWiP podcasts. It's it's a transformed portion of a striated skeletal muscle cell that Trichinella culls from a larger multinucleate cell and remodels it based on its protein secretions into a cell which maintains the life of this parasite for about 30 years inside muscle cells. And it's a true transformation. It's not a degenerative portion of the tissue or anything else. It's living tissue. It's got lots of structure. It's got a life of its own, basically. And I could recommend you going to my website for trichinella.org, and there's a pretty good description of the nurse cell biology there. And it's capital N because there are some nurse cells, I think, in Drosophila also that have been talked about because those are the cells, I think, that help the fly make eggs or something like this. I forget exactly what they're for. But well, I coined this term to indicate that the parasite needs help through its larval stages, and the host cell actually does that for it. And so the nurse cell is my sort of invention, I would say.
Okay, so I understand the question.
Yeah, right.
I get it.
The root nodule, actually, I thought I was going to go to root nodule nematodes, because root nodules and legumes and bacteria, that's a symbiotic association which is beneficial, right? That's a nitrogen-fixing type of strategy. But the root nodule nematodes, which actually subverts that process and actually kills the plants, forms a kind of parasite host cell syncytium at the contact point of the parasitic nematode into the plant cell root system. And it forms something that looks a little bit, not quite, but a little bit like a multinucleated nurse cell that Trichinella induces. And I've actually talked with those people about the similarities between my system and their system. And of course, they're concerned about this because there are a lot of economic reasons for being concerned about root nodule nematode infections of commercial plants like corn and soybeans and stuff like that. So it's a complex world out there. No matter which way you look, you're going to find interesting relationships that have very little explanatory biology associated with them that need more explanation.
Keeps us in work, Dickson.
It does. It allows us to shoot from the hip in terms of speculation and,
right.
Look like we know what we're talking about when we actually don't.
Thank you.
That's why science is the endless frontier.
No, I think you're right, Alan. I think we'll never know everything about everything.
All right, thanks, Dan.
Yeah, thank you.
Next one is from Niseen, who's a member of the Condit Lab.
Ah.
He sends us a link. He says, guys, do you think that if the water is contaminated with norovirus or rotavirus, the viruses will be killed with this method? They claim that polio titer is reduced by several logs. So this is a way called the SODIS method. You put water in bottles and leave it in the sun, and they say sterilize
The
it.
UV just knocks the DNA apart,
Yeah.
Right.
or the genome apart.
Now, I don't believe it because my experience with UV, it doesn't penetrate very far,
No, it doesn't.
right? So these look like 2-liter bottles here. And I would think...
Depends on what they're made out of, but you're right, I think.
They're plastic. I actually have read up a little bit about this in the past when it first came out. They're plastic, usually 1-liter bottles. The idea is to maximize surface area exposed to the sun and purify drinking water in places like Africa where there's no access to good drinking water supplies. And my understanding of it is that it gives you cleaner water.
Cleaner is right.
I wouldn't call it sterile.
No, I wouldn't either.
For a lot of the pathogens that are big problems in these areas, this could make a huge difference.
Yeah, but this isn't going to stop Giardia from being transmitted. And it's
Probably
not going to
not,
stop Entamoeba histolytica.
What about rotaviruses and noroviruses? I mean, it reduces the polio titer. Polio is pretty hardy. And they're similar. I mean, the noros are similar in genome
What about
size.
influenza
Also, the
virus?
killing is going to be twofold. It's not just UV light. The water's going to heat up.
Yeah, if you did it in the right place, the heat would probably work.
And as I say, this is intended for tropical countries especially, where clean drinking water is a huge problem.
You bet.
So if you put a 1-liter bottle of water like this on the roof of your, I guess the website has it shown on the roof of a grass-thatched hut, and you're somewhere near the equator, you're going to do a lot of damage to the microbes that are in there.
Dickson, they say Giardia and Cryptosporidia are killed by UVA even when the water is cooler. They say they've tested it. They've tested it against a wide
UVA,
range.
not UVB.
Yeah. So what is...
The difference, I mean, UVB is the frequency of the DNA molecule itself, and that's why it gets knocked apart and the dimers form. UVA has got a higher intensity, but it's at a wrong wavelength in terms of interacting with the DNA molecule, I thought.
So they have a table of all the pathogens they've tested. Bacteria, 99.999% reduction, 6 hours at 40 degrees.
Yeah.
Rotavirus, polio, hepatitis. 99.
Yeah, they've got a lot of. They do have a lot of. This is not BS. There's a lot of peer-reviewed research that they've done on.
Giardia.
I'm surprised. That's all I can say. I'm surprised.
Cysts of Giardia rendered inactive.
I'm surprised.
Cryptosporidium amoeba not rendered inactive. Water temperature must be above 50 degrees for at least 1 hour for amoebiasis.
So it's a temperature thing, not a UVA thing. Maybe the UVA heats up the water or something.
It's probably both. It's probably some of the UVA, certainly the surface of the water and also the temperature.
Well,
there must be some interesting taste too.
Right. Indeed. But when you're thirsty, what do you care?
This is recommended by WHO and Red Cross as a way to treat drinking water in developing countries.
Yeah, because they have very little else to.
We might as well do something that's low tech,
yeah,
right?
As I say, this is intended for places where the alternative is nothing.
Yeah,
correct.
And so if you can reduce the pathogen load a fair margin, and even if you're not getting them all, that's that's some people who live to see their second birthday. So.
Right.
Okay.
So I presume these plastic bottles are discarded Coca-Cola bottles.
Yeah.
Because they're
no shortage of them.
There's all over the place. Yeah.
Yeah.
So does
you can't get clean drinking water, but you can get Coca-Cola in a lot of places in the world.
You got it right.
SODIS stands for solar water disinfection.
And in fact, Coca-Cola Company has been very instrumental in getting vaccines delivered to the rural areas.
Well, I'm grateful, Nassim, for sending that to us.
Yep.
We'll put it in a link in. Victor writes, first, I want to thank you for the excellent podcast. I started from episode 1. I'm up to 80 plus now, enjoying every single episode along the way. I love the science, the stories, the personal touch, even the sound quality. But above all, I love learning about the whole new world of virology. I am a software developer, and I find it very refreshing to discover thought-provoking knowledge outside of software and computing that broadens my understanding of the world around us. I came across today a podcast episode that might interest you. It is an interview with the investigative journalist Brian Deere about his 7 years of reporting and legal issues surrounding the 1998 article in The Lancet claiming that the MMR vaccine causes autism and bowel problems. It is fascinating to hear the minute details around the history, the documentation trail, the litigation that led to the erasure of Dr. Wakefield from the medical register. I know most of the facts, however, this interview paints a very disturbing picture of corruption, dishonesty, and lack of moral judgment. And he puts a link to it, which we'll include in the show notes. Best wishes to you and the team, and keep up the good work. It is much appreciated. Victor is from London. Thank you, Victor. Not too far from the White Cliffs of Dover, Dickson.
Indeed, indeed.
Mm-hmm.
Kelly writes, excellent conversation with David Tuller. Many journalists do not understand science or only receive training in public health with no emphasis in the biomedical sciences. Vincent, you wondered aloud how many XMRV grant applications the Virology Special Emphasis Panel will receive for review. Apparently, it depends on the disease being studied. All CFS studies regarding XMRV are being automatically referred to the CFS and Fibromyalgia Special Emphasis Panel, where Myra McClure has been appointed to review those applications. Good virologist, specific point of view. Given that the CFS Fibromyalgia NIH SEP is funded at the same level of funds as hay fever, $4 million per year, with only approximately 6% of those funds actually going to CFS research in 06, 07, and 09, according to NIH figures, it isn't very realistic to expect enough funding to have sufficiently powered XMRV studies coming from this area. Do they automatically send XMRV applications involving prostate cancer to a cancer SCP?
I would expect they would.
So they have some standing study sections on cancer. They could go there as well.
Yeah, and I would expect the virology special emphasis panels are supposed to be for virology studies, not clinical research, right?
Yeah. I mean, in this case, there is no study section that really would be appropriate for CFS. It would always be mixed in with some other areas. So I think the idea is initially to give a special emphasis panel where you have experts, but of course, in this field that's so polarized, it's very contentious.
Right.
So anyway, I found a link to a composition of all the NIH study sections, and we'll put those in the show notes so you can see that. There is an NIH conference going on at the moment on CFS, ME/CFS, where they're trying to identify future directions to go in research. So maybe we'll talk about that in a few weeks. And that will do it for email. Amazing. Let's move to some picks of the week.
Excellent.
Dickson, do you have a pick?
I do. I have a specific pick this week. It's from Science Magazine. It's the latest issue of April 1st, 2011, volume 332. And the title of this article is Bacteria-Phage Antagonistic Coevolution in Soil. And it says that what you observe in a test tube between a virus, a phage virus, and its bacterial host equals a situation that you might find in an arms race type of situation where the evolution of one is then enhanced by the evolution of the other, so that it's a spy versus spy kind of thing of, the black spy versus the white spy in Mad Magazine. But in the soil where these organisms are ordinarily found, the situation is quite different, apparently, and the host cell appears, at least in short-term evolutionary process, to become more resistant to various forms of phages than you would get in an artificial situation where you don't have any other interfering things to think about, such as climate change issues related to temperature and humidity. So, it's very interesting because when you take this back into the real world, the situation looks a little bit different than it does in the laboratory. And we all know this, but here's a good example of one of those things that involves a bacteria and a phage, things that we're all used to talking about, but rarely do you find people taking these systems back into nature and finding out what they're really like. So I like this article a lot.
Cool.
Yeah, rarely do we take experimental results from animal models of virus infection and then repeat them in people. Right?
Well, that's tough.
Can't do that.
That's a tough one. I think Koch's postulates stops just short of that one.
Yeah.
The IRB,
these days, anyway.
Yeah, the IRB doesn't look friendly with things like that.
Yeah, by the way, people who are reading the other site that I plugged a couple of weeks ago, the Walter and Ina site, are going to see some insight into that pretty soon.
Oh, cool. I've been following that. That's great. You're so lucky to have those letters.
They're a blast. I'm
Amazing.
doing them about a week ahead, and just got through some really good ones coming up. So,
Welkin, what do you have for us?
My pick is actually the Walter Reed collection at the University of Virginia, and this is an online digital archive that I actually have blogged about before on Small Things Considered, but I thought I'd pick it here because I think it's especially relevant for the listeners of TWiV. And this is it's a vast collection of digitized documents and photographs and things surrounding the career of Walter Reed, but mainly the work he did in the very beginning of the 20th century, proving that yellow fever was a transmissible disease and it was transmitted by mosquitoes. And it's of interest because yellow fever is caused by a virus, and it's my understanding it's the first human disease that was specifically attributed to a virus. Because this was in the era where they were first figuring out that viruses even existed.
Yeah.
And the archive is really neat because for people who don't know the story, what they did is they went to Cuba where yellow fever was rampant. Walter Reed and surgeons from the Army and a team of scientists, and they basically did human experiments. They set up these huts, control huts, and then huts with mosquitoes, and they would put human subjects in these huts, so local Cubans, and so the archive is interesting because you can find everything in there. You can find handwritten fever charts from some of the patients who contracted yellow fever, telegrams back and forth between Walter Reed and other people, and what I think is the most interesting document is what the website thinks may be one of the first examples of informed consent, and you can actually see a scan of these original one-page documents that the team used to get the person, the so-called volunteers, I guess. Well, they got paid. I guess they weren't volunteers. That they would have to sign off consenting that they were being used in a human experimentation. And I'm going to read a passage. So what they would get is $100 in gold if they did this.
That's a lot of money then those days.
Yeah.
And actually an extra $100 if they contracted the disease.
Yeah.
And was that for burial fees?
Yeah. Well, so this is the paragraph. You can never get away with this with an IRB now, but the undersigned, so this is a specific one. It's got the signature of Antonio, I can't quite read it, but one of the individuals that participated. It says the undersigned understands perfectly well that in the case of the development of yellow fever in him, that he endangers his life to a certain extent, but it being entirely impossible for him to avoid the infection during his stay in this island, he prefers to take the chance of contracting it intentionally in the belief that he will receive from the said commission the greatest care and the most skillful medical service, which sounds a little bit like arm twisting to me.
Army twisting, that's right.
And it does say that if you die, that the money can go to somebody else that you designate.
I love this, that you picked this, because we have a plaque which is between the admissions committee office at PNS at our medical school, and the it's called the hole in the wall, but it's actually a little commissary where you can go buy some coffee and stuff like this. The brass plaque commemorates Jesse Lazier and Agramonte, who were both graduates of our medical school, as being participants in this experiment. And
actually, Lazier died.
They both died.
Yeah,
they both died, but the story is very interesting because Lazier died because his friend Agramonte actually performed the experiment of putting the little cup with the mosquitoes inside on the arm of Lazier, and Lazier caught yellow fever and died.
So Lazier's fever chart is in here.
Yeah, but Agramonte had the same thing happen to him. They did it to each other, and Agramonte didn't die. Any guesses why? But he died. I'll tell you why in a minute.
He did die or he didn't die?
He died, but he didn't die from yellow fever.
Oh.
Because he'd already had it as a child.
He'd had it, yeah.
So he went off and committed suicide.
Oh, I didn't know that.
Because he killed his best friend.
No.
Wow.
Wow.
Yeah. That's a very tragic story.
I didn't know that.
Yep.
Dixon, we'll have to go to the plaque and you can record a little.
I did already. I think I did a TWiV on this already.
No, we'll go to the plaque.
Oh, yeah. No, we'll read it. And you know what?
You can stand in front. I'll videotape you reading it. We'll
By the way, no medical student at this school, at our school right here, knows anything about that plaque until I used to bring it up in my class.
Cool.
And I used to say, by the way, on your way to the commissary, take a look at this plaque.
Next week we'll go over that.
Yeah, sure we will. We'll photograph it and put it up online.
We'll take a video.
We will.
Actually, the other thing you can see here is the telegram that was sent to Jesse Lazier's wife. It's like one sentence, and it just says that he passed away. No details.
Exactly. And by the way, Walter Reed was not present for those experiments. He was.
I don't think he
was back up in Washington, but
he was the PI.
Yeah, that's at the Walter Reed Army Hospital. I've actually seen the painting depicting Walter Reed watching the experiments being done, which isn't true at all.
So there are a lot of clippings here. I just clicked on one from 1901. It's from the Times, Topics of the Times. It says, Dr. Charles Finley of Havana evidently feels that he is not getting quite as much credit as he deserves for having definitely and publicly declared many years ago that the relation between yellow fever and mosquitoes was about what it has recently been proved to be by the brilliantly successful experiments of the government board. Isn't that funny?
I gave you a paper. It was a reprint of a paper by Finley on yellow fever.
Fever chart. This is great.
You know why he didn't get it right.
Why is that?
My understanding is those early experiments, the mistake they made is they didn't allow for the few days of incubation time for the virus that the mosquito fed on to replicate in the gut and go back. Whereas Walter Reed's group did that. They had mosquitoes that had fed on the blood and waited a few days. So they're infectious.
There's another interesting part of this, though, because they suspected all along that mosquitoes transmitted it. But why was it important to show which mosquito?
To know if the ones in the Panama zone were. Yeah. Why? What did they?
Which mosquito did they find was the transmitter of the yellow fever?
Aedes aegypti.
That's right. It has a special biology. Mm-hmm. It breeds in temporary bodies of water.
What do you call them?
Treehole mosquitoes. And so knowing that it's the treehole mosquito, it means you could develop a campaign against it.
Right.
If it was like Aedes or Anopheline mosquitoes that breed in open water, you wouldn't have had a chance in hell of interfering with their life cycle. But in this case, because it turned out to be the treehole breeder mosquito that was actually the transmitter, they could do something about it. And without it, they wouldn't have completed the Panama Canal.
So did they spray or?
No, they just
got
emptied.
rid of water?
Yeah, they just got rid of all these. The French tried to build this canal first, right? And then they gave up because it was too expensive and they had too many deaths. They looked at the debris. For instance, right now there's an expedition on Mount Everest to debride Mount Everest of millions and millions of pounds of discarded camping equipment and all kinds of climbing equipment. Well, the same thing happened at this site. Imagine what it's like for 30,000 people. You've got all their debris of these tins of food that they discard, and all the boots and everything else. All of those little places are places where this mosquito can breed. And simply by cleaning them up, they got rid of the yellow fever epidemic. And of course, it's also got a wildlife look-alike in the trees surrounding that, in the Haemogagus mosquitoes and in the monkeys. But as long as you don't allow them to come into camp, you can keep yellow fever from spreading into human populations that way. So they learned a great deal about a lot of ecological aspects of the transmission of diseases through these experiments.
It's nice, beautiful.
Yeah, I love it. I love
really
it. I love
great.
it.
Really
This
cool
is
stuff.
Fabulous,
I had
fabulous.
not known
Welcome.
this.
Thank you so much.
Thank you.
Nice. Who's next? Alan.
I'll go. My pick is a blog I discovered just a little while ago called The Artful Amoeba. And I searched through our past agendas so that I thought maybe we had picked this before, but apparently we hadn't.
I like how the show notes say Alan the Artful Amoeba.
Yes. Yes.
I guess I read that wrong.
Are we looking at Dictyostellum here?
Up at the top.
This one?
I think that
I don't know.
I think that may be
Fruiting
Dictyus.
bodies.
About the header image. Let's see.
Yeah.
It's the wasp nest slime mold, Metatrichia vesparium.
Oh, that's a
There you
slime
go.
mold of some sort then.
Nice.
Okay. Looks like a bunch of grapes.
Protists.
Yeah.
Yeah, so this is a blog by a science writer who is deep into biology, and she writes on a variety of biology topics. Looks like her most recent post is about paramecium, but then she'll do paleontology and just delves into all aspects of the field. And
that's neat.
Really fun.
She should take a look at what the chromatoidal bar of the cyst of Entamoeba histolytica looks like.
Well, some of the resolution
It's beautiful.
Is incredible.
Vince, it looks like this. Vince has on his desk these little magnetic balls, and if you let them assemble in a proper way, they look like a tobacco mosaic virus. If you look at the electron microscope picture of the chromatoidal bar of histolytica, it'll look just like this. Remarkable, beautiful symmetries in nature.
There's also a picture from Haeckel.
Yes.
All right. Sure.
I have a book of these.
I do too. I have the same book. I hope it's a reprint, though, right? Unless you.
No, I think I bought it at Woods Hole.
The original.
I keep it on my desk under the coffee cups.
You could retire any minute.
Haeckel's the guy I think that first did illustrated evolution as an actual tree.
Yeah.
The first phylogenetic tree was his.
Wow, this is good stuff.
This is beautiful.
Great stuff.
Yeah, it's a neat blog. So
Isn't this what keeps attracting us to science?
Yes.
It's the beauty underneath all of these things that really attracts us.
She is a science writer.
Yeah.
Very nice.
If you're interested in hiring me as a speaker, freelancer, or guest blogger, wow. We do it for free.
We do.
Yeah.
We do.
I guess that's her living, so.
Well, she's got a fossil one in here.
Yeah. Yeah, she
this
just
is-
ranges all over the place. I mean, I kind of range over a pretty wide territory on my blog, too, but she really delves into some neat stuff that I don't necessarily get to.
I thought I ranged over viruses, man, but this is even wider. That's cool.
I love this title. And this is how we culture cellular slime molds in Germany.
That's cool.
Slime?
Slime mold. Okay, my pick is an article in Microbe Magazine from the American Society for Microbiology. It is called Safety by Nature, Potential Bacteriophage Applications. It's an article from a professor of microbiology at the University of Florida and chief scientist of Intralytics, a biotech company focused on developing and commercializing bacteriophage-based products. So it's a little summary of using phages to get rid of bacteria.
Are you going to say his name?
Sure, Alexander Sulakvelidze. Sulak Velid,
pretty good.
something like that. Do you know how to say it?
No.
The reason I came across
We call him Alex.
Yeah, right.
The reason I came across this on TWiV number 4, one of the hosts, Michael Schmidt, mentioned this article. He was talking about using phage therapy in Russia to treat Salmonella gastroenteritis. And so he mentioned this, so I thought this was good, and it is an interesting little article. A number of listeners of TWiV have asked about bacteriophage therapy. So there you have it.
I hope they read this article in Science about how the phages and bacteria behave differently.
You know, Dickson, what the problem is?
What is the problem?
It's behind a paywall.
Oh, you're kidding me.
Yeah, you can't access it.
Science is behind a paywall. Yep.
Yeah.
You know what? We're going to scan this one and put it up.
Well, you wouldn't have to get the PDF, but that would be illegal, Dickson. And you don't do anything illegal as far as I can tell.
No, I try not to.
You're a straight shooter.
I am.
And that's the end of another straight shot of TWiV.
Yeah, I'll have one up.
Don't forget to visit our Facebook fan page, facebook.com/thisweekinvirology. You can find us at iTunes at the Zune Marketplace or at microbeworld.org/TWiV, and you can get their app for your iPhone or Android device so that you could stream episodes of TWiV to your phone. You can find it at microbeworld.org/app. If you subscribe in iTunes, do leave a comment. It's very easy to do, and it helps keep us on the front page of the medicine podcast section. As always, send us your questions and comments to TWiV at twiv.tv. Dickson Despommier can be found at verticalfarm.com, Racaniello.org, and medicalecology.com. Thank you for joining us, Dickson.
My big pleasure, indeed.
Always nice to see you. Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
Welkin Johnson can be found in Southborough, Massachusetts, which is affiliated with Harvard Medical School. He also blogs at Small Things Considered. Thanks for coming back, Welkin. He's gone?
Welkin is gone.
Oh, no.
Wait, we got to get him back.
No, Welkin just doesn't know how to use the mute button.
Okay.
Please don't leave us, Welkin.
I'm happy to be here. Thank you.
Hope you enjoyed it, and we'll come back again soon. Even when Rich is here.
Yeah, exactly.
I'm the president of the Rich Condit fan club now.
Very good. I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.